hide. Yes. Good. So, figuring out how to see you. It's not so easy. Now I can even see you. Okay, excellent. So let's get started. Today we are sort of swapping around screens. Yes. And I can pin myself so that you can see me. Yes. Right. So welcome to the LCI course. If there are some people also who are following who are not official students, and we are. So I'm going to start if I manage. Yes, here I am. So this is our facility. You have seen it yesterday, those who were here, and that's our building, and we are very conveniently located on both sides of this colorful bridge full of dichroic mirrors, or at least to me, it looks like dichroic mirrors. I really like this bridge. And we are in Sweden. So yesterday we had the first module where we were presenting our challenges. And today we're going to go to the module 2 and to start with, so working with light and fluorophores. And to start with, I'm going to give some key concepts of light microscopy. Then I have a few lectures, then again, some more key concepts. That's why it's called Key Concept 1. So I think it's important to have a sort of an overview of things, of the most challenging things in microscopy. And there are a number of them that many people do not know about, unfortunately, and this is the reason why they tend to think that microscopy is very easy. So segmentation is one of the keys to image analysis. It's not the only key, because there's some image analysis, some image analysis pipeline do not use segmentations, but it is one of the very important keys. So the goal of conceptualizing an experiment, or designing it, and growing animals and growing cells on the dish, and collecting patient samples and perfusing, cutting the sample, fixing it, staining it, and acquiring images, and then designing and running the image analysis experiments. The goal of all this is to extract numbers from the images so that we can compare them scientifically. And it's very important to keep this in mind. This is our goal, is to get numbers. So we have to get images that can give us reliable numbers. So classical image analysis is often made of several steps. There are other types of image analysis. As I said, for example, with AI and deep learning and all this, we have a new vast family of image analysis that opens up. But the classical image analysis is made of several steps, and one of the most challenging steps is called segmentation. So because I'm going to talk about this quite a lot during the course, segmentation this and there, I want to make sure that everybody's on board, and so I'm going to define this now from the start. Segmenting an image means separating the individual objects or areas in the image. So segmenting an image means that we're going to create a mask on top of the image, and in this mask we show which areas we're interested in, and outside the mask these are the areas we're not interested in. So this is, for example, an image of cells, and you have vesicles inside the cells, but you can see that we are interested in the vesicles, but we're not interested in that sort of haze that is behind, that is actually annoying us, whatever we want to call it, background. Anyway, it's there, and we do not want to analyze it. We do not want to get numbers from it. So what we do is that we make a segmentation mask, and there's like lots of techniques to do this. So we make this mask that shows only the areas we're interested in, right? And then when we overlay the mask on top of the image, then what we get is we basically get rid of the stuff we do not want, and we see the image as if these were like little holes, and you're seeing the image through the little holes. So these are the original data inside these, but everything else has been removed. This is the segmented image. So when I talk in the during the course, when I talk about segmentation, this is the process that I mean. Okay, so sometimes I will say, oh, it's much harder to segment the image. It means it's much harder to create the mask. Oh, it's much harder to, or it makes it easier to segment the image. That's what I mean. So then now you know. So when there are a number of concepts that are challenging when we do image analysis and when we do some microscopy. So in microscopy, the common microscopy artifacts, and we started talking about this yesterday, are refraction. So that is something that we really need to consider. Bleed through. We're going to deal with this today. Saturation and undersampling. This we're going to talk about a little bit later. Saturation and underexposure, sorry, and also undersampling. This we will talk about next week. And the resolution in the final image depends on several things. One of them, as I pointed out yesterday, is a big one, is the quality of the sample. And then we have, of course, the microscope. So the objective is extremely important for the resolution of the image and also the wavelength of the light that we are using. So all this we will talk about during the course. What I'm giving you now is like big picture thing. And then the resolution in the image also depends on the image pixel size. And this is something that is often forgotten or simply people do not know about it because they have never heard of it. Another key concept that is important to keep in mind is that you need to reverse think your experiment. So you need to start conceptualizing your experiment and designing, thinking image analysis. So that is the end step that people usually keep at the end. And that is a mistake because this is when you're discovering all the problems. So maybe that's why people keep it at the end because they don't want to sort of push it away. But this is what you really need to do this from the start. So you start with your image analysis, thinking image analysis, and then you design the sample accordingly. And of course, within the constraint of your sample, you cannot do just plain anything with your sample. And then you design the microscope settings in between. Most of the time, you will need several iterations of this. So you do not run the whole experiment and acquire all the images and then think about image analysis. So these are really important things to consider in microscopy. Something that is a little bit difficult to grasp, but it's really important. The image of an object is never, and it cannot be like the object. But we'll see this today. I think you will understand at the end of the day what I mean. What we image when we capture an image in microscopy is the interaction of the light that comes from the sample within the sample and with the microscope elements. So there's a lot of interaction of the light. That's what we are seeing. So I will explain to you today how the image is formed. And I think you will understand what I mean by the image of an object can never be like an object, the object itself. So we will now, this is the reason why I will now have a little lectures about the behavior of light and the properties of light. So I'm going to start now with the first little lecture. Nature of light. Let's have a look at what it is. So light is an electromagnetic wave. And so what does that mean? What is an electromagnetic wave? So basically, if we have a point charge, so it's a charge, but it's like a little point, and it's not moving, then it produces around itself an electric field. If it moves the charge, it produces still the electric field, but it also produces a magnetic field around itself. But now, if the charge moves with varying speed, which means it accelerates and decelerates, the electric field and the magnetic field, they become coupled. And they produce an electromagnetic wave which propagates through space. This is what the electromagnetic wave is. So light is an electromagnetic wave, and this is what it can be represented as. So we will have an electric field which oscillates. It means it increases and decreases, so it changes speed. And we have also a magnetic field that oscillates, and you see how they oscillate in unison. So they oscillate together, and that is an electromagnetic field. There is a change all the time, and that thing propagates through space. So it starts traveling. It has a travel direction. Okay, this is what light is. So we can represent light when we want to make a drawing of light. We can represent it in several ways. And I will use several of these during these little lectures today. So that's why I'm introducing them to you. So for example, this can be some light that is going through a barrier. Okay, here we have a barrier with a little hole and the light is going in that way and then it goes through. So we can represent light with its wavelength. So we in microscopy, we're very familiar with this because we talk all the time about the wavelength. But what it means, the wavelength is the distance between the crest. It's a wave, right? So it goes up and down. And so if we take the distance between the two tops, this is the wavelength of light. And it's in nanometers for us. In our field, and it is often represented by the letter, the Greek letter L, so lambda. Okay, so I will use lambda quite a lot in the course during when we talk about the wavelength of light. Another way to represent light is ray optics. So we can represent the direction in which the wave is traveling by a straight line like this, and that is ray optics. And another way to represent light is to draw a line on each of the crests, for example. And these are called wave front, okay? And you can see that they can have different shapes. So in here we have a flat wave front, whereas in here we have a curved wave front. And so these are the different ways to represent light that we will be using today. So light is an electromagnetic wave, but it also has energy. So there's some energy associated with it. And this energy is not evenly distributed along the wave. There is a much higher probability that it comes at different places. And this is like condensed. The energy is condensed into some sort of little packets, and they are called photons. So photons are actually the energy of light that is distributed along the wave. The electromagnetic wave range is very broad. So there are lots of different electromagnetic waves, and it's more than what we call light. So for example, if we look at cosmic rays and microwaves that you use in the morning to make your breakfast maybe, and the radio waves and the broadcast band, and all these are actually electromagnetic waves of the same nature as light. And then there is this very tiny little band in here. And if you blow it up, you can see all these colors. So this is visible light. And here we have UV and infrared, which we cannot see. But everything else is actually of the same nature. It's an electromagnetic wave. And what you can see is that we have, I go back a little bit. What we can see here is that these waves are very, very long. So they're very flat. They oscillate very, very slowly. You see here it's in kilometers. And here it's in like, I don't know, picometers maybe, minus 12, I can't remember. That would be nano. Okay, no, nano is here. So this would be pico, I think. So here we have nanometers or hundreds of nanometers in the visible range. So usually we go from around 400 or a little bit before to a little bit after 800. This range is extending now. We are more into the 800 nanometers than 700 like we had before. And so what we can see is that if you think about cosmic rays, gamma rays, X-rays, these are very damaging for us. If we get exposed to X-rays and gamma rays, this is very dangerous, right? Even UVs. But radio waves and the broadcast waves, they go through us without us realizing. They go through buildings, so they can travel. They are very long waves. So they do not see, let's say, the small opticals that we are. And they just go through us and we do not realize it. And when you think about this, you understand a little bit better that the more blue or the more towards the left side of the spectrum we go, this light is more damaging, whereas this one is less damaging because it's longer wavelengths, and longer wavelengths, they are less damaging for our samples. So basically, if we go towards the left side of the spectrum, we have light that is more energetic. So these photons, they carry more energy than the red ones, but this blue light is more damaging and it has a higher resolving power. So we will see this a little bit later. And unfortunately, it's stops very quickly. It is stopped very quickly by all the obstacles that it finds. Whereas this light in the longer wavelength is very penetrant, and it can go through things without being so much disturbed, but it has less energy, and it's less damaging for our samples, but has also a lower resolving power. Okay, so this is the type of choices that we have to make when we choose the color that we want. Do we go for more resolving power, or do we go for more penetration, for example, and less damage? So the wavelength of light determines how energetic, penetrant, and damaging the light is, and also how we perceive its color, and the maximum resolution that we can get in the microscopy image. And we will talk about all this during the course. You will be experts. So now I've been talking a little bit about the resolving power. We have to define something else that is important. So what is resolution? The resolution is the shortest distance between two objects that can be identified as being separate. So if we have two objects, but we see them as one, they're not resolved. If we have two objects and we see that there are two of them, then they are resolved. Now I'm saying C, clearly we don't want to look at it with our eyes. So the question is, can we separate them with image analysis? And for that, this is a whole field because we can separate them with so many different ways. So when we have, so that is a little bit tricky, but this, because the resolution is the distance between two objects, it means if we have a high resolution, a high resolving power, this is a short distance. When we can say that it's two objects when they're actually very close to each other, we have high resolution, okay? So it's a little bit sometimes tricky to think about it, but when we have a high resolution, the distance in nanometer is getting smaller. And another word to define that can be useful is isotropic resolution. And isotropic resolution means that we have the same resolution in X, Y, and Z. So in the 3 dimensions of space, same resolution is called isotropic resolution. So this is the visible area of light, and we're going to focus on that clearly because this is what we're talking about today. So the visible light is between more or less like a little bit before 400 nanometers to about 800, more like 750, I think is the maximum where we can see. And here we have all the different wavelengths depending on the color. So you can see that they're becoming smaller. This is 400 nanometers and this is 700. And here you can see all the different colors. White light, when we shine white light on our sample, white light is a mix of all these colors, or a number of them. But basically, white light is not a pure color, it's a mix of color. And we can have different light sources that we use in microscopy, and they emit a different range of wavelengths, but they also are not even in their distribution. So you can see here, for example, a mercury lamp will emit across the whole spectrum, but some wavelengths are much brighter than others. So this is what the range that the mercury lamp emits. It's very uneven. Whereas lasers are very monochromatic, so they're going to emit a few nanometer and a very much one specific color. LEDs, on the other side, they have a bit broader, they are sort of in between. So they are a broader range. So very often with LEDs, we actually need excitation filter to make them narrower. And their range is in around the 50 nanometer, 60. Sometimes then we have to use an excitation filter, but we will talk about all this. Okay, now we would like to talk a little bit about the behavior of light. So when we often say light scatters, what does that mean? If light travels in homogeneous matter, it just travels straight without being disturbed at all. But clearly our world is not homogeneous, and our samples are definitely not homogeneous. So we now are working with light in heterogeneous matter. So the particles, all the particles in our sample that are larger than the wavelength of light that we're using, they scatter light. So they split it and send it in all different directions. Scattering is actually a combination of reflection, refraction, and diffraction. This is a real mouthful. to say, and I will explain what this means. But scattering is basically a general word to say, I'm exploding my light. And it leads to the spreading of light energy. And so that means that when the light that is scatters arrives at the sensor, we're recording a lower intensity, and also we have light a little bit everywhere, so everything is hazy. So this is what we mean when we say scattering. So if you look at this picture of beautiful Sarek National Park in Sweden, you can see mountains. And in here, you can see all the details of the little bushes. And it's very clear that all the mountains have these little bushes on the lower slope. But in here, in the faraway mountain, you cannot see these details. And what you can see here is that the light that between me and this taking the picture and this mountain, there is more air, and this air contains water particles, dust particles, lots of things that will scatter the light. And so when it reaches me and I can take the picture, then I have the light that comes from this area, I have more scattering, and therefore I have less contrast and less colors. So this is exactly the same thing that happens in our samples. Here, if I'm close to the cover slip, I see lots of details, and then I'm far away, I don't see a lot of details. So this is what we are trying to avoid when we prepare our sample. So in fluorescence microscopy is exactly the same thing, but one has to realize that both the excitation and emission lights are scattered in the sample. So we have double trouble. So all these short wavelengths, the shorter wavelengths encounter more particles than the larger wavelengths. So if you think there is like a certain range of sizes of water droplets between me and this place, then all the short, you think about the big buildings, you know, the long wavelengths, they just pass the building. They don't even see the buildings. They don't see me. They just go through me. But the short wavelengths, they are blocked by everything because so many things are bigger than them. So it's a question of the relationship between the object and the size of the particle. So the short wavelengths in this white light encounter more particles than are larger than them, and therefore they scatter more. Whereas the long wavelengths in light are less disturbed, and so they penetrate more because they are able to travel undisturbed for a longer distance. So let's have a look at, sometimes we can see, we talk about light is being absorbed. So what does that mean? So the samples also absorb light, and different materials absorb different wavelengths, and they absorb differently. And the wavelengths that are not absorbed by the material, they escape from it. And this is what we see, because then they reach our eyes or they reach our sensor, and this is what we can see. And that's how we see their color. So this is an example of the beautiful Swedish archipelago. I'm doing a little bit of PR here. And so if you can see here, the sunlight is yellow, so it's full of lots of different wavelengths. And some of this light is reflected on the surface of the water. And what we are seeing is that the light, we will see now reflection, how that works, but the light is not going through the water. This light is going straight into my eyes or into my camera. And therefore, I have the same color here. So there has been no change of light. It just bounces off the surface of the water. However, the light that is here, this light that I'm capturing with my camera has gone through the water, and part of it has been absorbed, and everything that is not blue has been absorbed. And this is the reason why only the blue light comes out, and this is what I'm capturing. So the water looks blue. So this is the sunlight that has been transmitted through the water, and it is not, this is whatever is not absorbed. That's what I'm taking a picture of. So this is the difference with absorption. Now, I used to have, when we had the course live, I used to have a torchlight with me, and torchlights, you know, they shine white light, and I would put the torchlight behind my hand. But so I just took a picture. So this is my fingers, and then there's the torchlights right behind. And what you can see is that even though the torchlight shines white light, actually what comes through my hand is red. And that is because my blood absorbs all the blue and green light. And so what is left after you've removed blue and green from white light is red. And so this is what goes through. And again, the red is a long wavelength. So it has managed to go through my fingers, and then this is what I'm imaging. So light scatters only on objects that are shorter than its own wavelength. And in tissue samples, the short wavelengths are absorbed faster and they scatter more and they warm up the sample more. And this is what makes it damaging. So all this heat in the sample. So the conclusion is that if you want to image live samples, you should use a long wavelength if you can. You're going to have a little bit less resolution, but this will be much better for your sample. So all this behavior of light has consequences for us as microscopists. So the conclusion from this little lecture is that light is an electromagnetic wave, and we have wavelength. We can represent it as wavelength, rays, ray optics, or wavefront. And the visible range of the image that we collect most of the time is between 400 and 700 nanometers. And the short wavelengths are less penetrant, so they are blocked quite quickly in biological tissues. They're more damaging, but they have a higher resolving power. And scattering happens on both the excitation and emission, and we would like to try to make our sample as little scattering as possible. And so we lose the intensity in the image and we lose the sharpness. Good, so now we're going to do what we will do during the whole course. So we're going to have a little quiz. So it's a very short quiz, so we can stay in this Zoom. And I'm going to stop sharing. What you will do now is that you will go to Canvas, and I will share my Canvas so that people who are following can see it as well. So you go to Canvas and you go to the first module, and you will see there okay, right now, click. Okay, now it works. So in the first module, you can see, I can share that. Look. You will see the quiz, and then you just do it. So this one, share. Sorry, I'm half to screen, so I look on the left side. So here we go. We have quiz, nature of light. So you just do that. You work it on it individually. I can show the screen quiz for people. Show the answers that are possible. Pick. Okay, now I go to the second question. And when you guys are finished, we're going to have a little break. So we will be back at 10 for the next lecture. Okay, I will stop sharing now. I think that people have done their way. So I'm just checking that indeed this is the case, but yes, exactly. So we will be back at 10. So see you guys in a short while. If you have any questions, you're also, of course, very welcome to ask me now. So we're going to do these sort of quizzes throughout the course. And you have seen that when you answer the quiz, you get the answers that I would give. And I think that is also a way for you to learn. So we'll do zillions of quizzes.
Hi, Sylvie.
Yes.
I have a question of
Yeah.
the quiz because I have finished the quiz and even the answer is correct, my score is 0.
Oh yeah, there's no grading for that.
Okay.
Yeah, it's these are practice quizzes, and you will see that sometimes I forgot to remove the points because when I add a question, it puts points automatically. In any case, even when there are points and there are no points, they are not counting towards the final grade. So
Okay, thank you.
yeah, you know, it's my I haven't had time to change all this, to change them to 0 or whatever. I just want to tell you that all these quizzes, they are for you to practice and for you to be able to formulate. You know, now you have a drop-down menu, but many of these quizzes you actually have to write the thing. So it's very important for you to formulate in your head and be able to express the answer to the question, and then you will see the way I would have answered. But these quizzes do not count for the final grade, even if they are 1 or 2 points or something like this, because I haven't managed to remove them. I think that you can see at the start. Can't you see at the start? It's written. Maybe it's not written. Okay, sorry. I thought that you would be able to see at the beginning. Does it count for the final grade or not? But maybe you don't see that. So I can tell you, all the small quizzes, they don't count. But they're very important for you to learn.
Sylvie?
Yes?
Are we going to talk about the answers of the quiz later, or if we have questions now, we should?
Yes, please. If you have questions, ask me.
With the RNA detection in fixed sample, I understand the concept of the shorter wavelengths is going to give a better resolution, but in the end of your talk, you said that long wavelengths have like less scattering and more penetration.
Yeah.
So we balance these things.
Exactly. Unfortunately, this is the way what we have to do all the time.
Okay.
So we have to balance these things. So basically, between resolution and with scattering, and you know, if it's the thicker samples, for example, if you have a thin sample and you're looking at RNAs, so you don't care too much for an RNA stain because it's not going to be live. So you don't care too much if it's damaging to the sample or not, but you care if it's scattering. And if you have, now it's possible to do RNA stains in thick samples. And so it's becoming very important. So, you know, we have to balance all these things. This is what we will have to do all the time during our life, I guess, as a microscopist, is to balance things.
Thanks.
Welcome. Are there any more questions? So if not, I see you guys at 10 in the same Zoom.
Hi.
Yes?
It's me, Yufei.
Yes.
In my left cell staining kit, they provide the whole set rather than the which one? DRAQ5. So, I mean,
yeah. You mean for the first question, right?
Yes.
Yes. I see what you mean. So you mean that you have Hux or you have DAPI and you have no choice. Is that what you mean?
I have only a whole set. I tried the DAPI as well, but it seems it doesn't work.
What do you mean by the whole set?
the hottest.
Sorry, so you have a kit, is that what you're saying?
The kit is what itself includes a nuclear staining dye, and it's
Cells. It's cells that include the nuclear dye. No, I didn't understand.
I mean,
You're using a kit.
Yes, itself. It contains a nuclear dye.
Yeah, exactly.
And
So
it's